I'm Takezawa from Japan. I'm going to talk about the spinal neurons processing bladder sensory input of mice are different from those of rats and cats. Spinal neurons projected from bladder afferent nerves are considered to be involved in processing of bladder stimuli. However, little is known about which spinal neurons process which bladder stimuli. The aim of this study is to identify the spinal neurons which are projected from bladder afferent nerves and involved in processing of bladder stimuli. Study design. We used male black 6J mice. We performed two experiments. Number one, a neural tracing, and number two, spinal shear force induction study. First, we performed neural tracing. We injected bilateral neuron tracer CTB into the bladder wall. 1 week after injection, we analyzed the distribution of CTB in the L6 and S1 spinal cord. Here is the results. CTB positive nerve fibers were distributed to lateral dorsal horn, sacral parasympathetic nucleus, and dorsal commissure. Next, we performed spinal shear force induction study. To provide mechanical, inflammatory, chemical, and acid stimuli, we infused saline slowly, saline fast, LPS, ATP, and acetic acid into the bladder for 2 hours. We analyzed C-force induction in the L6 and S1 spinal cord immunohistochemically. Here are results. As you can see, regardless of the stimuli, C-force positive cells were distributed in SPN and DCM. These results suggest that the mechanical, inflammatory, chemical, and acid stimuli to the bladder activate the spinal neurons of SPN and DCM in mice. This finding is not consistent with those of rats and cats because noxious stimuli to the bladder have been reported to induce C-force positive cells not only of SPN and DCM, but also of medial dorsal horn in rats or lateral dorsal horn in cats. Conclusions. The spinal control systems for voiding reflex are considered to be different among mice, rats, and cats. Thank you for your attention.
It's fair to say that you've looked at the afferents, but how do you know this is the input to the voiding reflex in your conclusion here?
Sorry?
Well, your conclusion is saying the spinal control systems for the voiding reflex are considered to be different. Now, I agree you've put in, there's an afferent pathway demonstrated, but that looks like the pain afferent pathway because you've used lipopolysaccharide and important noxious stimuli, which aren't the normal afferent limb of the voiding reflex.
It's correct.
I'm not disagreeing this is important information. This is important information, but it's just your conclusion is a bit odd.
Thank you.
Any comments from floor? So I think that we have to really pay attention to this finding because mouse is such an important model. Rat is such an important model. Pain is so badly understood. We have so many studies that are saying important conclusions and extrapolating to human. And it could be that with this important insight, you're explaining to us that extrapolating to human from some of these studies is a bit unwise.
Yes, I think so.
Yes, thank you. No other comments from the...
I agree with you. You need just to be sure about this, because otherwise all experiments are not usable for us.
No, I think this is important, but you should be careful. This is not the voiding reflex input. I think it's the pain input, and that's a very important pathway. Thank you very much.
Thank you very much.